Alright, hello everyone. Today we're going to talk about mutations. By the end of this video, you should be able to tell me what the 3 types of point mutations are, and you should be able to tell me the 3 consequences of point mutations. So let's get started. I want to quickly recap protein synthesis. Remember, protein synthesis is where you go from DNA to RNA to protein. This step where you go from DNA to RNA, that step right there is called transcription. And the step where you go from RNA to a protein, that is called translation. So very quickly, if we wanted to transcribe this piece of DNA that I have, let me get yellow here. If we wanted to transcribe it, C would pair with G, G would pair with C, G would pair with C, T would pair with A would pair with U, T would pair with A. That sequence right there, that's what I've written in this box because that's our piece of RNA. We just transcribed the DNA into RNA. Then, every 3 letters, and this is very important, every 3 letters of the RNA represent a codon. And that's why we have this table to the right, because that is our codon chart. So if we wanted to look up what amino acid is coded for by these 3 letters, GCC, we would look in our table and find G, C, C, find the place where all 3 meet, and all 3 meet at alanine. So that's why I wrote alanine there. You can practice on your own if you want with the next codon, AUA. If you look at it in the table, it makes isoleucine. Okay, that's a recap of protein synthesis. Here's why I mention protein synthesis. If a mistake is made in the DNA, if there's a mistake, then that mistake can show up in the RNA, and when you make a protein out of it, that means the protein that gets made could be mistaken as well. It could have mistakes in it. It could code for the wrong things. And when we talk about these kinds of mistakes, we're talking about mutations. Okay, so a mutation is going to be a change to one or more of the bases in the sequence of nucleotides. Okay, so right here I have a sequence of nucleotides. Now you're only seeing the bases, those 4 letters, A's, G's, T's, and C's. But remember, the nucleotide is made up of a sugar, a phosphate, and a base. If I change one or more of these letters in the DNA, then that's going to end up changing some of the letters here in RNA, and that could cause a change in the protein as well. And so these kinds of changes to one or more of the bases we call a mutation. So there's going to be a specific type of mutation, 3 types of point mutations. So when I say point, I'm just talking about one of the letters. Okay, so we're just looking at what happens when we change one of the letters. There's 3 types of those mutations. The first one is called, let me make this a little, the first one is called substitution. Okay, and if you compare this piece of DNA to this piece, you may notice there's a difference. The letter A here has changed. It was in our original DNA, it was the letter G, but now it's been changed to an A. But notice we still have the same number of letters total. There's 6 bases there, but we've substituted this G with an A. So that's an example of a point mutation that we call substitution. The next type of point mutation is called insertion. like you're inserting something. Insertion. Okay, so insertion is when you take a base and you just add it to the sequence. So you're not going to swap something out like we did with substitution. You're just sticking another base in. So you'll probably notice that the letter A there that I've highlighted in red, it has been inserted between this C and this G. That's where that A went. And we still have all the same letters that come after that, but we stuck an A between the C and a G, and the G. Because of that, it caused all the other letters to shift over one place. And when all the letters shifted over one place, it can cause some serious problems. So just to kind of give you an example of how this would look if we were looking at it in English. If I had the sentence, the dog can sit, notice every one of those words is made up of 3 letters, just like our DNA up here. Every 3 letters is going to be our what makes our codon. If I insert the letter Q between the H and the E, if I insert the letter Q there, then that's going to cause the E and all the other letters to shift over one place. And if that happens, it gives you a sentence that doesn't really make any sense. And so this, you get an idea of the how big a mistake an insertion can be. It can cause a lot of problems. There's also a third type of mutation, and that is called deletion. Deletion. So this is where we take one of the letters in our original DNA, and one of them gets erased or removed. So we still have our C and we still have our G. There should have been a G next, but this G right here got deleted. And so once it got deleted, the same problem happens. These next 3 letters shift back a space. Okay, and I'll give you an example of a sentence down here that does this. So if I had the dog can sit and I deleted this E, then all the other letters would shift back and you get a similar result. The sentence now doesn't make any sense once we deleted that E and the other letters shifted back. So insertion and deletion, we say that those can cause a frame shift. Okay, those 2 types of point mutations can cause a frame shift. And by frame, I'm just talking about these 3 letters. Every 3 letters, that's our reading frame. And so if you insert a base or delete a base, it can cause the other letters to shift either forward or back, and that changes the frame. All right, those are the 3 types of point mutations. And then you can have 3 consequences of a point mutation. So, for example, this is our original piece of DNA here, TTC. Well, if I take that C and I substitute it for a T, then we can figure out what the result would be, what the consequence of that would be, just by comparing what would happen if there were no mutation and what would happen now that we have a mutation. So let's see what happens. TTC, when we transcribe that sequence into mRNA, it would make AAG. And then when we translate AAG into a protein, it's going to give us the amino acid LYS, lysine. All right? But what happens, let's compare that to what happens when we take that C and we substitute it with a T. Well, then our mRNA is going to be AAA. It was AAG. That would have been normal. Okay, it was AAG. Our mRNA is AAA now, and when we look up AAA in the table, it gives us lysine. Well, that is the same amino acid that we would have gotten even if there weren't a mutation. So if you get the same amino acid that you would have gotten if there were no mutation, then we call that a silent mutation. When you get the same amino acid you would have gotten if there were no mutation at all, then we call that a silent mutation. That's one consequence that you could get of a point mutation. Now, a different consequence, I'll erase this, a different consequence could be if you substituted the letter T here in our original DNA with the letter A. If you do that, then follow the same steps. Our mRNA would be UAG now. Okay? If there weren't a mutation at all, it would have been AAG still. But now it's UAG. And when you look up UAG in our codon chart, so UAG, when you look it up in the codon chart, it codes for a stop. So whenever there's a mutation, a point mutation, and it leads to a stop codon, we call it a nonsense mutation. Okay, so that's another type of consequence of a point mutation. It could cause a nonsense mutation where we get a stop codon. All right, and then last, you could take our original piece of DNA here, TTC, and for that T right there, we substitute it with a C. Notice what happens when you do that. Now our mRNA becomes AGG. It should have been AAG. And when we look up AGG in our codon chart, it leads to arginine, ARG. Our amino acid should have been lysine, though. So whenever you have a point mutation and it leads to a different amino acid than the one you would have gotten, we call that a missense mutation. Okay? So those are the three consequences of point mutations. You can either get a silent mutation as a result, a nonsense will always lead to a stop, or a missense mutation, which just leads to a different amino acid. And that's all for mutations.